Good morning, good afternoon, and good evening, everyone. This is Liam Sanyo from Inside Scientific, and I'll be your host for today's event, which is titled Quantifying Cardiovascular and Behavioral Correlates of Fear in Mice, Implications for PTSD and Cardiovascular Disease Risk. This webinar has been sponsored by DSI, Colburn Instruments, and Harvard Bioscience, so a big thanks to them for making this event possible. Today we're being joined by Dr. Paul Marvar, Associate Professor in the Department of Pharmacology and Physiology at George Washington University, and Benjamin Turley, who helped to conduct the research presented today as an undergraduate student in Dr. Marvar's lab, but is currently a master's student in systems medicine at Georgetown University. Today, Paul and Ben will present research pairing behavioral and cardiovascular responses to cued fear learning and demonstrate how fear-based disorders like PTSD can contribute to cardiovascular disease risk. And with that, I'm very pleased to welcome Dr. Paul Marvar and Benjamin Turley. Thanks so much, Paul and Ben, for being with us today. And the floor is yours whenever you're ready.
Perfect. Thanks, Liam. I appreciate the introduction. Ben and I are really excited to be here to present some of our work regarding quantifying cardiovascular and behavioral correlates of fear in mice. And I'd just like to thank Inside Scientific and the sponsors, Harvard Bioscience, and the opportunity to present today. Yeah, so there are basically three learning objectives that we'd like to achieve during this webinar. First, we're going to be reviewing a little bit the physiological impact of PTSD on cardiovascular and autonomic homeostasis and how this can contribute to cardiovascular disease risk. We're going to demonstrate the utility of some of our recent studies using rodent models that builds off of previous earlier literature in which we're able to assess real-time cardiovascular and autonomic physiological measures of fear or defensive emotional states. Finally, we're going to briefly discuss the benefits and translational implications for using integrative behavioral and cardiovascular multi-modal methodologies, in particular how they can advance the understanding and the intersection between neuroscience and cardiovascular disease. I'd like to actually just acknowledge the work that I'm going to be presenting is largely a result of work by Dr. Adam Schwartz, who's now at the National Institutes of Mental Health as a postdoctoral fellow, continuing in this line of research in the context of hippocampal dependent learning processes and integrating telemetry-based approaches in rodents. And finally, Ben Turley, who's now a master's student at Georgetown, performed a lot of these studies, the latter part of the studies, which we're going to be discussing, or I should say Ben is going to be presenting. So I'd like to start out just briefly with the question, why do we study behavioral and cardiovascular correlates of learned fear in mice? Well, as this little cartoon indicates, there's increasing attention and research to brain, heart, body relationships, to mental well-being and physical health. And this is becoming more and more apparent today. And in the context of living through a pandemic, I should say. And then I want to say that with regards to my laboratory's research interest, which is focused on understanding the link between anxiety disorders, such as post-traumatic stress disorder and cardiovascular disease risk, the intersection between brain, heart, body relationships is an important component. And our laboratory is focused on understanding these mechanisms at a basic science level. So what is PTSD? PTSD occurs in about 8% of the population following a traumatic event. And this could be a personal assault, natural or human-made disasters, serious traffic collision, or military combat. And I should add that in veterans, the rate of incidence of PTSD can be as high as 30%. So PTSD is characterized by a maladaptive reaction to exposure to severe psychological trauma. And it includes various components. There's psychological and behavioral components or symptoms. And these symptoms occur about 3 months following the traumatic event. And these could include intrusive recollection of that traumatic event, avoidance behaviors, negative mood alterations, which may include heightened anxiety, anger, depression. And I also want to point out that there's a lot of physiological correlates or symptoms that an individual PTSD displays. This includes hypervigilance, physiological hyperarousal, increased blood pressure, heart rate, disturbances in the autonomic nervous system, such as increased sympathetic outflow, respiratory distress, shortness of breath, and sleep disturbances. So there's a large physical component that we're going to be talking a little bit more about with regards to understanding that from a basic science level. And I want to add too that with regards to PTSD, currently there are limited effective treatment options. Cognitive behavioral therapy is one type of effective option that individuals typically respond about 30% of those with PTSD. And then antidepressants in terms of the pharmacological options are a common treatment option. So our research in particular, we're hoping that some of this work could lead to more effective options of treating some of these different physiological as well as psychological symptoms. And I want to highlight too that the PTSD is highly comorbid in terms of poor physical health outcomes. And this includes both, in addition to cardiovascular disease, metabolic and autoimmune disorders as well. So there's increasing evidence in both population and epidemiological type-based studies that are showing this link. And this, in terms of these physical health comorbidities, this has become an increasingly large problem as it was recently identified by the Centers for Disease Control with regards to the link between anxiety, depression, chronic stress, post-traumatic stress, and how they contribute to poor cardiovascular, cardiometabolic outcomes. And as shown in this schematic, there are bidirectional relationships. So there's some clinical evidence to suggest that the mental health component leads to poor cardiovascular and metabolic outcomes. And then there's also evidence to suggest that conditions such as stroke and heart failure, metabolic disease, or I should say an incident, for example, having a heart attack itself could contribute to post-traumatic stress disorder. And so with that, understanding these mechanisms, what do we know? We know that there are physiological pathways that include both pain, heightened fear, increased cardiac reactivity, reduced blood flow to the heart, and increased cortisol levels, part of the fight or flight response. So these are important physiological abnormalities that can occur that can contribute to these bidirectional relationships, as well as behavioral or modifiable pathways, which include smoking, physical inactivity, and medication non-adherence. So understanding these connections between anxiety disorders and cardiovascular disease risk is an important public health concern. Back in 2018, the National Heart, Lung, and Blood Institute brought together a number of different scientists, clinicians, and I was proud to be part of this effort to really understand or look at, identify some of the current knowledge of this research, identify some gaps in understanding and potential future research opportunities. And I just want to draw the audience attention to this recent publication that describes some of these different components in terms of the understanding between post-traumatic stress and cardiovascular disease risk. And it really highlights the importance of the multidisciplinary approach from both an animal and basic research perspective, which I'm going to be talking to you about today, as well as epidemiological based research, genomic medicine, and clinical and translational type research, which is important to help with or advance current treatment options. Okay, so in terms of PTSD contributing to cardiovascular disease risk, so some of these physiological and behavioral elements that I've discussed are nicely shown here by a review in Lancet Psychiatry by Donald Edmondson of Columbia. And as shown here, following trauma or PTSD, this figure here really describes what represents the strong evidence shown in blue, as well as the emerging evidence for this PTSD link shown in orange. And one of the areas that we're particularly interested in is high blood pressure or hypertension, as well as dysregulation of the autonomic nervous system. Inflammation plays an important role in many disease states, and not surprising that following trauma and PTSD, there's heightened inflammation, which is thought to also impact the elements of central control of blood pressure, as well as peripheral outcomes. So in terms of, I should just add here, in terms of the outcomes, acute cardiac events, there's strong evidence for that stroke, as well as formation of blood clots, which is interesting. There's emerging evidence for this as an outcome, cardiovascular disease outcome following trauma and PTSD. So I mentioned that high blood pressure is one of the areas that we're interested in. There's evidence to suggest that individuals with PTSD following trauma, that they have elevated resting blood pressure, in particular in military veterans, as well as community-based samples. So greater resting blood pressure, so reactivity to trauma-related cues is also an important component. So individuals that may recall that traumatic event or associate a specific cue to that event are known to have enhanced reactivity of blood pressure, as well as autonomic control or sympathetic outflow mechanisms. So I'm talking a lot about the brain, and I think it's important because in terms of PTSD, the brain, and there's the brain, which as shown here in this schematic, is impacted by PTSD in the context of there's specific regions that are involved that become impaired. There's cortical regions, which are important in detecting and perceiving the threat, such as the visual cortex, which projects to the hippocampus, which is important in contextualizing that threat, as well as the amygdala, which elicits or contributes to the behavioral and physiological responses through the brainstem. And then in PTSD, the amygdala is thought to be hyperreactive, whereas the prefrontal cortex, which somewhat serves as a brake on the amygdala, has been shown to be hypoactive. And this relationship, or these circuits, are critical with having an appreciation and understanding for how some of the behavioral, physiological, and cardiovascular responses may go awry, such as increased reactivity to threat cues with blood pressure and heart rate. And this classic fight or flight response, again, the impaired ability to process fear and perceive threats is a major component to PTSD, and usually what drives these hypervigilant or hyperphysiological responses that are seen in patients. So in terms of the evidence from a clinical perspective, as I just mentioned, the sort of impaired ability to process fear and perceive threats, what does that mean? That usually is indicative of overgeneralization of trauma-related stimuli. So a differentiating between a safe versus a dangerous environment or cue. And it's the individual's reduced ability to suppress that emotional stress or physiological response. And in the context of this talk, we're going to be talking about blood pressure, heart rate, sympathetic nervous system, and threat-associated stimuli. A collaborator at Emory University, Jeannie Park, has made some really important observations in veterans with PTSD when they're exposed to virtual reality combat, VRCE. And then she's also identified that these individuals have heightened, elevated blood pressure, and that when in response to certain stressors, such as VRCE or other descript stressors, these individuals have heightened sympathetic reactivity. And also, she's identified that the baroreflex, which is important in controlling blood pressure responses to stress, is impaired. So the idea that PTSD is not limited to psychological symptoms is not new. And this is a paper back over 100 years ago describing or making the observation that veterans of war exhibit abnormal cardiovascular responses that are associated with trauma. And they refer to this as irritable heart syndrome. And what did they find? They found that these veterans exhibited heightened heart rate and respiratory responses to presentations in civilian life to bright flames or general pistol shots. But understanding this component of the physiological component to the psychological trauma is an important area that often gets overlooked in terms of treatment. So having a more mechanistic understanding using animal models, we and others propose that is required for a more comprehensive understanding of the neurobiology, including neural circuits, autonomic dysfunction, neurotransmitter abnormalities, as well as heightened inflammation. So I'm going to talk to you a little bit about the models we use and how we're looking at these from a basic science or neurobiological mechanistic perspective. And so Pavlovian fear conditioning is a widely used model used in both rodents and humans to study fear responses. So we're able to model a fear response in both rodents and humans. And the basis for Pavlovian conditioning is essentially pairing two stimuli, typically a light or a tone, which is the conditioned stimulus, with the unconditioned stimulus, or the aversive stimulus, which is a foot shock or air blast. And when you pair those two stimuli together in an experimental context, when they co-terminate, this is referred to as associative or fear conditioned learning. And when the threat stimuli is then presented following this association, typically 24 hours following, there's a robust fear response. And this is sort of defensive or avoidance behavior, freezing behavior, enhanced autonomic arousal, reflex or startle potentiation, elicits a strong stress hormonal response, such as increasing in cortisol, as well as respiratory distress. In terms of the model, this is how it looks like. In terms of the lab, the mice are paired with an audible tone and a light foot shock. And that light foot shock happens on day one, and this creates the fearful association. And then the animals are then put through 24 hours later what's referred to as extinction training. So extinction learning is a process that's impaired in PTSD. So individuals with PTSD are unable to diminish or inhibit or weaken that association. And this occurs in experimental contexts. We're able to test this through multiple repeated exposures to the conditioned stimulus. And you can test this in rodents over multiple days. For example, on day 3, we can test the recall of that weakened association. And so freezing behavior and avoidance. These are classic ways in which neurobiologists and behavioralists study this fear conditioning response. And as you can see, when the animals are fear conditioned, their freezing response increases. And the freezing response is an evolutionary conserved physiological response that is important both rodents and humans in terms of it's a state of immobility, and it's designed to avoid the perception of a threatening situation. And so you could see what happens with over time. The animals learn that the aversive stimulus is no longer aversive, and their extinction learning increases. So this is enhanced extinction learning, enhanced extinction testing over multiple days. So I mentioned this is impaired in PTSD. So an individual in the context of an impaired extinction process, you have heightened freezing. So in our laboratory, not only do we integrate a component of a physiological component of defensive responses or learned threats using freezing behavior, but we also use integrative approaches to measure the cardiovascular activity. And this is using both heart rate ECG signals as well as blood pressure using wireless telemetry. And so what does that look like in terms of experimental study? So the mice are implanted with a DSI HDX11 transmitter that it's put into the carotid artery of the mouse and then subcutaneously implanted. The mice recover 7 to 10 days later, and then we're able to monitor their cardiovascular and autonomic state and activity, body temperature in a home cage environment. And then the system is integrated with software known as Panema, which we're going to be talking, Ben's going to be talking to you about a little bit more later on. And we're able to record from up to 12 mice in a scheduled session or in a continuous session in terms of collecting data from both the heart rate and blood pressure channel. So the blood pressure channel, you're able to acquire both systolic and diastolic blood pressure, which is allows for the assessment of mean arterial pressure. What's unique is that with the ECG data from the heart rate, the ECG channel, we're able to further look at potential defects in autonomic signaling in the mice in response to threat cues, and that's through using time domain and frequency domain analyses. Assessments of heart rate variability are possible. These are both measures of autonomic nervous system function. So next I'm going to kind of put you through, go through a couple studies that we've conducted using this paradigm. And the first study here, we wanted to evaluate the effects of extinction learning on the conditioned cardio-autonomic response. So I mentioned these are studies that Adam Schwartz conducted, and he did an outstanding job with this paper. We have this paper in there if you want to learn more about this particular study. So in this study, we hypothesized that a fear-conditioned cardiovascular response could be detected in a novel context. So the novel context, for example, is this context A, context B conditions, and attenuated by extinction learning. Okay, so the animals are habituated initially to the fear conditioning environment. We go through 2 days of habituation. And what we did was we recorded blood pressure first at baseline, and then we subsequently measured blood pressure and heart rate following fear conditioning and following extinction training. And then we measured these responses during extinction learning, which is what we wanted to look at with the context of understanding the cardiovascular response with regards to extinction learning. So this table here, I just want to highlight, there's 2 groups here. A group that received the conditioned stimulus only, so the CS only, and the conditioned stimulus plus the US. So that's the fear conditioning group. But what was determined from this data was that the animals that went through fear conditioning, their basal resting blood pressure was unchanged by that procedure itself. So fear conditioning itself did not change resting blood pressure levels. And this is shown by your light dark cycle variations in this table. So next, what we wanted to do was measure these responses in the extinction training context. So following fear conditioning, the animals acquired fear, as you can see in panel B, where you have heightened freezing response. And then in panels C, F, and G, essentially this is the integration of and time synced freezing, mean arterial pressure, and heart rate response. So animals that were fear conditioned, you can see following the first CS presentation. So there's 30 CSs that were presented during this time period. So you have the no CS period and then CS 1 through 30. And you can see a classic freezing response indicative of the acquisition of learned fear. And this is an expression of fear following 24 hours following fear conditioning. And the other thing I want to point out here is the mean arterial pressure and heart rate are nearly at reaching a ceiling effect here, in terms of physiological limits in the mouse. As you can see here, nearly 800 beats per minute. A mouse's heart rate is normally at around 550 beats per minute. So you can see this is a very heightened, aware mouse that's exploring the cage and exhibits a strong freezing response. Now I've highlighted here some important points with the blue arrow. You can see during the first CS presentation, we see a small increase in blood pressure, but what's notable here is we see a reduction in heart rate. And this is seen in humans as well, and this is what's referred to as a fear bradycardic effect. And this is due to enhanced parasympathetic stimulation on the heart. And it was really interesting to be able to observe that during this experiment and during this context. So you can see the other thing that I want to point out is following the no CS period, where the animals, their freezing behavior stops, and what you see is this rapid increase in heart rate. So that was the second observation that Adam noted that was quite interesting, which could be in effect due to their increased activity, but clearly these are important cardiovascular physiological measures that we're seeing in conjunction with the freezing behavior. So in the same context, 24 hours later, we then looked at how well the mice learned in terms of the freezing response. And you can see that in panel D, their freezing behavior is reduced, suggesting enhanced extinction learning over that period. However, the blood pressure response, we still saw this increase in blood pressure, which the blood pressure then stayed stable during that particular extinction training session. And then what was really interesting too is that heart rate itself, you could see over that period in both the CS only group and the CS US group, there's sort of a habituation effect with heart rate that we also noted. And then you do see a little bit of a bradycardic effect in the fear-conditioned only animals, again suggesting that the actual fear conditioning itself is providing some unique unique contributions to autonomic function compared to the CS group only in terms of the fear bradycardic effect. And then this data, E, G, and K, I'm not going to go through that, but that's data where we averaged both day 1, day 2, and the early extinction versus late extinction as well. And so these are the couple points I want to highlight in terms of extinction learning as well. So one of the things that Adam noted was that because of the high basal state of the animal in the extinction context or the novel context, it was difficult for us to really get a true conditioned reactivity, cardiovascular reactivity response in these animals. This is due to the elevated baseline cardiovascular measures. And so in the second study, we wanted to examine the effects of extinction training on cardiovascular reactivity in a home cage environment. And that is shown here in terms of the difference between activity and cardiovascular measures between a training context and a home caging context. And you can clearly see that in the home cage, shown in black, the animals are in a resting state. So they're not moving, they're nesting, they're in a resting state. We next wanted to see what effect that the conditioned stimulus had on the animal in this particular state. And then again, to look at the extinction learning effects in the home cage context. In terms of what that experimental paradigm looked like, so all of the animals were fear conditioned on day 1. They then went through a home cage test, which consisted of 4 conditioned stimulus stimuli that were 30 seconds each. And then the animals either went through a extinction learning paradigm, which included 35 CS presentations, or no CS, but exposed to the environment. And so these two groups then were tested immediately following extinction learning. And then we tested long-term extinction learning and how these effects affected, or how the extinction training affected the animals long-term. Okay, so this is in terms of test one. What you can see here in the panel, both upper and lower, is that the animals following fear conditioning in response in the home cage environment, in response to the conditioned stimulus, you see a robust increase in both groups, black and blue, in response to the conditioned stimulus. And then upon the second stimulus, you see a steady increase and sharp rise in that blood pressure and heart rate response. And this persists over the next 3 to 4, and then that response begins to diminish over time. And then the heart rate response, we saw a general increase in the heart rate response, but in terms of how the pattern that we observed in blood pressure was not present in the heart rate response. And yeah, so that's that. And there. So in terms of extinction training, we then went out ahead and put the animals through extinction learning and then tested them with a 4CS test. And what Adam found was that when the animals were exposed following extinction learning, when they were exposed to the conditioned stimulus, the animals in blue, extinction learning animals, the response compared to those that did not was significantly diminished. Although we did see a slight increase in blood pressure on this first testing day in response to the first 2 CSs, you can see that. The response was significantly attenuated. And the heart rate response, although not significant, we did see this made this observation that the heart rate response on that first testing day was not elevated. Then on day 3, so this is sort of long-term recall of the conditioned memory response. This is the 3rd testing day. And you can see that response compared to the animals that did not go through extinction learning is significantly attenuated. So this is shown here. And then what are some of the take-home points and conclusions that we drew from this study? Was that resting blood pressure, heart rate, and heart rate variability or activity levels were unaltered by fear conditioning and extinction training. So the resting blood pressure, cardio-autonomic state was unaltered. However, when we put the animals through extinction learning, so recall of a consolidated extinction memory, we found that this process modulates the conditioned cardiovascular response, which is influenced by context-dependent differences in blood pressure and heart rate. And we saw that the blood pressure response, which was much more consistent and reproducible than the heart rate response. And concluded that the conditioned blood pressure response may serve as a novel index in the evaluation of extinction efficiency and may aid in further understanding mechanisms that drive exaggerated physiological responses in PTSD. These studies are ongoing. I would also like to highlight that collaborators at Harvard, Antonio Saligowski and Kerry Ressler's group, are looking at these cardiovascular responses, and Tanya Jovanovic, who's now up at Detroit, they are sort of at the forefront of looking at cardio-autonomic responses with regards to fear learning and trauma-exposed patients. And I'd like to point out and highlight this particular paper, which made some interesting observations between early and late extinction and the differences in the cardio-autonomic response. Okay, so with that, I'm going to turn it over to my colleague and contributor to this work, Mr. Ben Turley. And he's going to talk to you a little bit about some of the challenges that we faced with this model and some of the things that we've done to circumvent it with a particular software that we think, application that we think is an important next step for some of this work.
Thanks, Dr. Marvar. And thank you to everybody for setting this up. So the same problems with telemetry in general are also going to affect the retrieval of learned fear. This is especially the case when we're using cardiovascular measures of behavior. There are a lot of things that can affect the cardiovascular state of the animal. Environmental factors, cage setup, and whether the animal's close to other mice. And also the physiological state of the animal is determined by circadian rhythms of routine. In fact, we found that blood pressure response to acute stress is going to be affected by the circadian rhythm. So just sampling at different time points is going to affect the data collected. Similarly, there are going to be some practical limitations to conducting measurements using cardiovascular telemetry, considering that the mouse diurnal schedule is effectively opposite of our own. And I'm definitely not going to be staying up in the lab at 2 a.m. to make sure that we're collecting good data. So that and the factors noted above make the collection of cardiovascular data difficult, as the response might be modulated by the physiological state of the animal. We tackled this problem using software, specifically Ponemah, which Dr. Marvar mentioned, and it's built to control the conditions during which we can elicit a cardiovascular response using a conditioned stimulus. So Ponemah itself is going to be sending data to the computer, like traditional telemetry, where we record vital signs, heart rate, blood pressure, but it also has the secondary mechanism where we can send TCP packets to this software. And the software is able to act on those signals in real time and dynamically adjust a conditioning schedule based on the instantaneous cardiovascular state of the animal. So the software is able to run a conditioned stimuli without an experimenter present and will adjust based on the cardiovascular state of the animal to make sure that the animal is not tested when it's in a period of abnormal activity, as defined by the experimenter. We tested this using the same HDX11 transmitters, which we used before for conditioning and retrieval, and these were subcutaneously planted. We also wired speakers directly into the home cages, and these were all controlled by this software program. The animals were fear conditioned, and the software was configured to give 6 conditioned stimuli over 24 hours, which is the only case to my knowledge where conditioned stimuli were able to be delivered both day and night. We also specified that the mean arterial pressure, MAP, had to be within the parameter range, in this case 85 to 100 millimeters of mercury for 10 seconds before the CS was delivered to ensure that the animal was in a reasonable cardiovascular state. These are the specific parameters used. These are the ones exposed for the experimenter. And you can see here that there are a couple functions which were not used. There are a couple native functions which were built in. For example, you can use multiple speakers. Each can have their own dynamic tone schedule. And you can also shift the parameter range up during the nighttime to account for diurnal presentation. But one thing worth noting is that the minimum interval length here was set to 120 minutes. And the reason for that is because the software is going to be pushing the tones back based on the cardiovascular state of the animal. So if the animal is not in that parameter range, it's going to wait until it is. And that creates the problem where if the tones are presented too close together, it might affect the response. And we also want to make sure that the animal is not going to get extincted in case giving the conditioned stimulus several times in a row without the unconditioned stimulus is going to change that training. So this is going to enable state-dependent conditioned stimulus presentation. This is a graph of one of the tones presented. And you can see, first of all, the gray shading where the tones actually peaked into the parameter range. And the computer didn't actually give the CS the tones during those times because we had set that it has to be in the range for a certain amount of time. And this actually proved useful in this case because you can see that the mouse started off at a blood pressure where we determined it would be unacceptable or the data would be too soft to actually work with. It started off around 75 millimeters of mercury. And you can see at the points where the blood pressure peaked into the parameter range, it actually declined again. So this was useful because we gave the tones at a point where the mouse was actually rising and staying in an acceptable range around 550 seconds after the scheduled time. And then the tone was delivered there. And you can see that in this live trace. It might make it a little bit easier to see when the tone was given. Here we have a figure of all the stimuli presented. And right off the bat, one interesting thing we found with the ability to deliver tones at any time of day was that the points where the animal was in its resting period, blood pressure response to the tones was actually delayed. And I find this funny because it's probably the result of waking up a tired, groggy mouse, but it's important to note the differences in behavior based entirely on time of day. Because if you're delivering tones later into the day, when the mouse is in its resting period, and you're using the same heuristic, the same measurement, which is to check the difference from baseline a couple seconds after the tone, it might not actually be valid when the mouse is in its resting state. Because you can see here, there's actually a delay of around 40 seconds before we see a cardiovascular response. So these things are important to know, and I think it'd be good to tease this out with a larger sample size. Another thing worth mentioning is that CS6 was never actually given during the experiment. And the reason for that was because the mouse never actually entered the range that we considered would be acceptable, the experimenter-defined parameter range. And lastly, as Dr. Marvar mentioned, we are collecting heart rate here, but it was not used to demonstrate fear response as it would be in people. And this is simply a difference in species, that blood pressure has been found to be a more reliable indicator of learned fear. And I think overall, this is showing the need for a dynamic baseline when considering condition response. Okay, I'm just going to wrap up here about the main benefits of the software and then some of the other functions it can use, which are pretty interesting. So overall, the main benefit means the experimenter doesn't need to be present to deliver the CS. And it opens the door for giving tones at several times of day, which were previously difficult to do with a normal working schedule and generally never used before, despite the fact that it's during the nighttime that the animal's active period would be most similar to our own. It also allows the experiment to happen in the animal's home cage, where the animal might be less stressed overall. There are a couple aspects of the software which could be useful for behavioral response quantification. For example, other behavioral measures like respiration are sent with this data packet, and it can be used in place or in conjunction with blood pressure. The software is also open source. It's nothing fancy is going on in the background. It's made with vanilla JavaScript. Anyone with a little bit of programming knowledge can modify it. And it means that if you have a programmable piece of hardware, like a drug delivery apparatus, you could set the program to talk to that very simply to push a gas or liquid, or maybe just to notify experimenters an event is occurring. And also, one native thing that is built into the software is because you can set this parameter range in a custom way, you can set it to deliver tones explicitly when the mouse is outside of the normal range. For example, you could set the parameter range to be lower than where it usually is, and that would only trigger when the animal, for example, could be sleeping. Okay. Overall, I hope that this software is able to shed some light on the mechanisms relating to learned fear and learned cardiovascular response. And I hope it's able to make the lives of the experimenters who use it a little bit easier. Now I'd like to turn it back over to Dr. Marvar for the remainder of the presentation.
Great, thank you, Ben. I'm just going to wrap up with the sake of time, so we have time to answer some questions. So I'm bringing it back to understanding brain-heart-body relationships to overall mental well-being and physical health. So we think this work feeds into a better understanding for interoceptive processes and disease, and using applications like these. So interoception, which is the internal state, sensation of one's internal state, And the applications with regards to PTSD, I'd like to draw the attention to this nice review article from Maddox et al. In 2019. Identify the cardiovascular signals, which could be thought of as an internal state-dependent event. So increased cardiac signals and how they feed back to the brain, for example, and baroreceptor sensitivity, heart rate contractility. All these things are beginning to emerge as potential physiological factors that contribute to PTSD. So sort of the threat-dependent cardiovascular signal and an influence on PTSD risk and or cardiovascular disease development. So we think this is an important component that feeds into this work. And also these applications can be used in looking at different psychopharmacological drugs and experimental applications, as well as in animal models of cardiovascular disease, which is an important component to this work as well. And then one could envision the chemogenetic and optogenetic integration with applications like this that would really provide a sort of systems-based approach, multidisciplinary approach to understanding the influence of the cardiovascular system on PTSD and vice versa. So with that, I'd like to really acknowledge the people that contributed to this work. Ben, who's sitting next to me, but importantly, Adam Swartz, who did a remarkable job with laying the foundation for this work. Adam's now a postdoc, as I mentioned, at the NIMH, and he's integrating both telemetry and behavioral in his behavioral studies, but looking at both hippocampal-based mechanisms and understanding how the cardiovascular response and the role of the cardiovascular response in those type of studies. Current lab members, thank for their support, and then ongoing collaborations shown here, and obviously the funding that contributed to that work. So with that, Ben and I would like to thank everyone for registering for this event, webinar, and we'd be happy to answer any questions. And if there's questions we don't have time for, feel free to send me an email, and I'll be happy to entertain those as well. So, and thank you again to Liam and the organizers for this opportunity.
Thank you very much.
Fantastic. Well, thank you so much, Paul and Ben, for the really great presentation and for sharing your work with us. All right, moving right on to the Q&A. And let's kick things off here with a question from Lisbeth, who's asked, were you able to collect physiological data during the conditioning phase?
Yeah, that's a great question. And we did collect physiological data during the fear acquisition or the fear conditioning phase. However, during that period, what we observed is there's essentially a ceiling effect. So the amount of physiological arousal as measured by the blood pressure and heart rate response is nearly maxing out. So we didn't actually utilize that data in terms of our overall goal of that particular study. But it is technically feasible to do that if that was something that you were interested in. But again, the responses are, you reach a ceiling effect in that context.
Fantastic. All right, next question here was asked near the start of your presentation from Jessica. Are rates of comorbidity of PTSD with cardiovascular and other diseases higher than in non-PTSD individuals? So I know you touched on this a bit, but maybe you could just summarize.
Yeah, so in terms of that question, if I'm understanding correctly, so that the rate of PTSD with comorbid cardiovascular disease risk, are they different than individuals that have other comorbid conditions? In terms of, are you referring to other mental health conditions?
I think So rates of comorbidity of PTSD with cardiovascular diseases and other diseases higher than in non-PTSD individuals. So I think, yeah, comorbidity with cardiovascular diseases and other diseases, if they're higher in those with
Yep.
PTSD.
Yeah. So basically, I would say to that is that in general, comorbidities with other neuropsychiatric diseases, such as depression um, or anxiety, are higher in terms of the impact on the cardiovascular system. But the connection or link between there's a strong association, which I described, that's shown by evidence in both epidemiological and clinical studies for this connection between post-traumatic stress disorder and increased cardiovascular disease risk. And when I say cardiovascular disease risk, that can mean stroke, heart failure, coronary blockage, coronary disease. So, and that's what the evidence of the point that I was trying to make with that data.
Fantastic, great answer. Next question here comes from Jeff, who's asked, did all mice respond similarly to fear conditioning? And was there a percentage of animals that did not show a response?
Yeah, that's a good question. In general, the mice acquire fear at a similar rate. So that fear conditioning is a very strong conditioning event. And however, there were some animals that either did not their extinction or their freezing response to the condition cue fell below a certain percentage, which I think it was, in terms of when we tested this, below like 30%. I think those were data that we felt like the animal did not acquire fear or did not express fear at the level of what the majority of mice do in this situation. And the same goes for the conditioned blood pressure response. But in general, what we found is that there is a robust response that occurs in the majority of animals, but you do on occasion find outliers within these groups with this response.
Fantastic. Here's a great question from John, who said the time frame appears too short to be relevant to PTSD research, because people suffer from PTSD for months or years after the trauma. Do you know if there's any animal models or maybe methodologies that might take this consideration into account?
Yeah, that's a great question. And so in terms of the application that we're using this in, we're able to test both the acute within session, within the extinction learning, the acute 24 hours and 3 days following. But certainly you could envision using this application with other different behavioral paradigms that resemble symptoms of PTSD, such as passive avoidance, but also modifying your paradigm to where you can reinstate fear over a chronic period of time in an animal model, and then look at those effects chronically on the cardiovascular system. So that would entail, again, modifying your behavioral protocol to look at it more in a chronic way. And also, you could, from a behavioral model perspective, there are animal models that, and when exposed to severe stressors, that their extinction learning response is impaired. One in particular, single prolonged stress, or chronic unpredictable stress. These are conditions in which the extinction learning process is impaired, and again, resembles some of the physiological and psychological components of PTSD. And so one, again, could look at those parameters in terms of how they impact the cardiovascular system over time. So that's that. Hope that helps.
Yeah, fantastic. I think in the interest of time, we'll just have one last question, which comes from Matthew. The question here, yeah, have you examined whether the conditioned cardiovascular response varies between males and females, whether in mice or in human clinical research?
Yeah, so that's another excellent question. Thank you for that. And this scenario, I'm not aware of any studies that have looked at the sex differences in the conditioned cardiovascular response. It's something that we're starting to look at for important reasons. Merely the fact that in PTSD, clinical PTSD, women are almost twice as have a 2 times higher rate of PTSD risk and development than men. So this is clearly an important clinical problem that, and then the impact on the cardiovascular system. Women more at risk cardiovascular disease. So these things clearly will be an important component to look at. Again, I'm not aware of current literature that has looked at it from a basic science perspective. There's emerging clinical data that's starting to look at this work, and yeah. So, but yeah, that's where we're at currently with that question. But it's a great question. Definitely, there's a need for greater research in that area.
Yeah, certainly. Really interesting avenue for research. So thanks so much, Paul and Ben, for sharing it with us today. It's been a pleasure having you with us.
Thank you, Liam,
Thank you.
and everyone else that's contributed to putting this webinar together. Really appreciate it.
Yeah, certainly. Big thanks also to the audience here for taking part in this Inside Scientific webinar, and we look forward to having you with us again soon. Have a great day, everyone.